Hello everyone, and welcome to this podcast. In this podcast we're going to talk about DNA replication as it relates to eukaryotes. So we just got done talking about DNA replication in bacteria. And many of the things that we talked about with bacterial replication also applies with eukaryotic DNA replication. For instance, in eukaryotic DNA replication, we still need to be concerned about the new strands being anti-parallel. So that means we still have that leading strand and lagging strand. DNA is still read 3' to 5', and new DNA is still synthesized 5' to 3'. Many of the same types of enzymes are used. So for instance, we still need to have a helicase come in and pull the strands apart. We still need single-stranded binding proteins. We still need a topoisomerase. Again, that's the one we use. That's a slight difference here, is that in eukaryotes they don't use a gyrase, they use a topoisomerase, but essentially they're the same. They have the same function. DNA polymerase still requires that 3' hydroxyl, so we still require this RNA primer needed for DNA polymerase. And likewise, you still need the ability to remove the RNA and then to ligate those Okazaki fragments with DNA ligase. So many of the same kinds of enzymes are still being used. Now, even though there are many similarities between bacterial and eukaryotic DNA replication, there are some additional problems eukaryotic cells encounter. And as I continue with this podcast and talk about eukaryotic DNA replication, I will highlight some of those problems. However, I'm not going to go over every detail again because the basic process of replication in eukaryotes and bacteria are very similar. So I'm not going to redraw the replication forks with the enzymes involved and all that good stuff. I'm just going to highlight some of the problems that the eukaryotes have to deal with. One of the first problems, we can maybe number it here, the first problem here is that there's a lot more DNA in a typical eukaryotic cell. E. coli has about 4.2 million base pairs. Yeast, which is a single-celled eukaryote, has 16 million base pairs, so it's 4 times as big. Fruit flies, like you worked with in the lab, have 45 million base pairs. And humans have 3 billion base pairs. E. coli, as we've said before, has a generation time. It copies its DNA and makes new cells in 17 to 20 minutes. Humans, a typical cell will take about 24 hours to divide, and in that time they will copy their DNA. To go from 4.2 million to 3 billion, if you had a single origin of replication, it would take you much longer than 24 hours. It would take you perhaps months to copy the human genome. So as we've said before, eukaryotic cells, and I'm just going to draw a generic eukaryotic cell here. If we started replication here at one end and just worked all the way down, that would be a very easy way to do it. However, as I said before, it would take way too long. So in order to accomplish copying this whole genome in an efficient time, it's necessary to have multiple origins of replication. In this way, the cell can copy small parts of the genome at the exact same time. In fact, if we were to draw this out, let's just draw kind of a really long piece of chromatin here. At the exact same time, we may have a replication bubble here, 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 and here. Allowing DNA replication to occur at the exact same time at each of these bubbles, still going in a bidirectional manner, but at multiple replication bubbles at the exact same time. This allows the DNA to be copied in a much more efficient manner. Now one additional problem that this creates by having multiple origins of replication is, how does the cell know first to start replication at these origins and then to prevent multiple rounds of replication from happening at this replication site here, origin of replication site? Meaning once this DNA has started being synthesized in either direction, what prevents further rounds of replication from happening at that same site? As we begin this discussion, I want to have you recall the cell cycle that you learned about in previous courses, and we'll talk more about this as this semester goes forward. But remember, the cell cycle is the process where you take a cell, it enters this cell cycle, and it goes through this phase called G1. At G1, the cells are getting ready to have their DNA copied during S phase. S phase is when DNA replication occurs. And you can think of G1 as something on the lines of prepping for replication. And then it goes into G2, and then it goes into M phase. In G2, it's making sure that the chromosomes that have just been copied are prepared to go into mitosis, and in mitosis, those chromosomes are pulled apart to generate 2 new cells. And each of these cells go on to do their specific jobs or to re-enter the cell cycle. I mentioned this just a moment ago, but let's write out these 2 problems here that the cell has to encounter. It has to know when to copy, and it makes that decision, so to speak. A cell doesn't decide, but it makes this commitment to enter S phase right here. And you don't want to enter into S phase too soon, because it's possible you may have DNA mutations that have to be fixed first, or the cell may choose just to die instead of be copied. So it's very important that the timing of when DNA replication occurs is set. And the other problem is, we want to make sure that once replication starts, it is only copied one time. So once DNA replication starts here during S phase, it stops at S phase, and as the cell continues through the cell cycle, the DNA is not copied anymore. So remember these 2 rules. It has to know when to copy it, and the fact that it can only copy the genome one time. So I'm going to erase this just so I can draw the solution to this problem. Oh, you know what? Actually, I should have kept the cell cycle there. I'm going to redraw that. Next, I want to draw a part of the chromosome, and I want to just show maybe 2 replication bubbles here. Remember, the first problem was that the cell needed to know when to start replicating the DNA. And the cell does this by this very large complex of at least 14 different proteins. Your book calls it the pre-replication complex, or pre-RC. Some books call it, and I'm going to abbreviate it here, the replication licensing factor. Replication licensing factor, or RLC. Now what this pre-replication complex will do is bind to these origins of replication. Now remember, I said it's important to know when to bind. And so this binds after mitosis during G1 but before S phase. So I'm going to put a little star right here, meaning it has to bind before S phase. And it's highly regulated so that it binds knowing that the DNA is ready to be copied. So I'm going to put it right here before we enter S phase. Now once it binds, it's important to remember a little bit about what it's going to do next. If you recall, I said that it contained about 14 different proteins. One of these proteins is our good friend helicase. The same one that we talked about in bacteria that rips the double-stranded helixes apart. And so that's what it does too. As this binds here, the helicases in this complex begin moving down the double helix, ripping it apart and creating more DNA in a bidirectional manner with their leading and lagging strands. So this answers the first question. How do we know, how does the cell know where and when to start? It knows where to start because it's the origin of replication. It knows when to start, which is right before S phase and after M phase. Now there's this other protein that's really important. Some books talk about it, some don't, but we're going to talk about it because it's really important that the cell knows not to recopy this area, that once it's been copied, don't copy it again. And there's this protein called gyminin. And as soon as pre-replication complex binds here and the helicases start moving in opposite directions, ripping those hydrogen bonds apart and allowing DNA replication to proceed, gyminin binds here next, after this has gone on its separate ways. And so what you end up with is this origin of replication that's already been copied containing this huge gyminin protein complex. It sort of acts as a roadblock so that additional pre-RCs can't bind. It covers up the origin of replication so it can't recopy it. And so this binds at the end of S phase. What has to happen after M phase, because remember after M phase you've created a brand new cell here, and at some point you're going to copy the DNA. And so once this new cell is formed, right before it enters G1, gyminin falls off. So I'm going to go over this again real quick. As a new cell is produced, it enters into G1. Through various checkpoints, the cell is determined to have to be ready to copy its DNA. At this point, the pre-RC complex or the RLF complex bind to the origin of replication, allowing synthesis of the DNA or replication of the DNA to proceed. Once it binds, replication occurs. After it has started replication, gyminin binds to these origins of replication so that additional rounds can't occur. While as long as gyminin is bound to this origin of replication, additional rounds of replication cannot occur. Gyminin stays bound to this origin of replication all the way through G2 and all the way through M phase. Once the new cell is produced at the end of mitosis, gyminin is released from those origins of replication so that the pre-RC complex can once again bind here to do another round of replication. Okay, I'm going to go ahead and erase this. The next difference I want to point out here, so I'll put a 2, is that bacteria have 5 DNA polymerases. We only talked about DNA polymerase 1 and 3. Remember, 3 was the one that copied the DNA, and 1 was the one that removed the RNA primers. The other 3, you're not responsible for knowing, but they play a large role in DNA repair in bacteria. In eukaryotes, there are at least 15 DNA polymerases. You're not going to need to know all of them. If you look at your note-taking guide, I listed 5 that you needed to know. These include DNA polymerase alpha. This is similar to DNA polymerase 3 in bacteria. It initiates DNA polymerase with the DNA primase, meaning it needs that RNA primer. I also said you need to know DNA polymerase, gamma, and remember the mitochondrial eukaryotic cells also has its genome, and so it requires a different polymerase. And so this is the mitochondrial DNA polymerase. And then you need to know DNA polymerase epsilon. This is important in repairing damaged DNA. Also DNA polymerase sigma, and it's involved in DNA repair, similar to DNA polymerase epsilon. And the last one you need to know about is DNA polymerase eta. And this particular DNA polymerase is important in fixing DNA damage, but more specifically UV damage. That's what it fixes. So of the 15 DNA polymerases, you need to know these 5. And think about the roles as described here, but also think about what would happen if any of these DNA polymerases in the eukaryotic cell were mutated or non-functional for some reason. So I'm going to erase this now and talk about our third and final problem we want to talk about. And that is, how are we going to deal with these linear ends? And let me explain why this becomes a problem. Let's first draw a simplified version of bacterial DNA replication at the end of it. So remember, to copy this, you would have had to have had a primer, and then DNA polymerase 3 from bacteria would have bound to this and copied the DNA. And it would have got all the way to the end here, and DNA polymerase 3 would have come along and removed the primer and filled in this little bit here, and then DNA ligase would have come along and fixed that one last bond. So circular DNA is much easier to copy, because you just have to go in a circle until you get to the end. Eukaryotic DNA creates a problem. And what I want to do is draw one of these ends. So here's the original template DNA, 5' to 3', 3' to 5'. And I'm going to focus on the end down here. And so let's just assume this goes on far off our screen of view here. As DNA is being copied here, you have your primer set down there, and then you have your new DNA coming along, being copied. And again, this is going to be copied in the 5' because to 3'. Remember, the rule is it reads the template 3' to 5', and the new DNA is laid down 5' to 3'. And this goes all the way to the end and it stops. Then this RNA primer is removed and new DNA fills in that gap. And then DNA ligase comes along and finishes it off. So this strand down here, the leading strand, is no problem. We'll put no problem on this end of the molecule. Now remember, on the lagging strand, and why don't we just write lagging strand up here so we don't forget? Its DNA replication is discontinuous. And again, it's going to read in the 3' to 5' direction and make DNA in a 5' to 3' direction. So I'm going to go ahead and just lay down all the primers and then fill it in with the DNA extending from the primers. Leaving that small little gap right there. This is where we come into a problem. As these primers are removed on the lagging strand, And we'll save this one for a moment. New DNA replaces the primer, and DNA ligase comes along and seals this last bond, as we see here. So far no problem. Now remember, I forgot to put the orientation here, so remember this is 3', and this is 5'. Again, it's anti-parallel with the template strand. So what's going to happen now is that primer is going to be removed. The reason we can add DNA to here and keep moving this direction is because we have that 3' hydroxyl group that DNA polymerase requires. Right now what we have here is this gap here. We can't put more DNA here because the DNA polymerase doesn't have any 3' hydroxyl group to use to add new DNA to this, new nucleotides to this. So it's stuck here. Without having any downstream sequences, it can't fill this gap. So what happens at these ends now is that they now become unstable. Again, remember I told you before that a single strand of DNA is sticky. And now if we have a whole bunch of ends, they will begin to stick to each other. And you can end up with larger chromosomes that have bound to each other. If that doesn't happen, in general these become very unstable, and they can begin to get shorter, and that end is degraded. By itself that's not a huge problem, but then every time this is copied, this telomere, this end here, is going to get shorter and shorter. And eventually that's going to cause the cell to die. As this telomere gets, this end of the chromosome gets shorter and shorter and shorter, it will signal the cell to die. Now in a moment, I need to explain two things. First of all, I want to explain how the cell accomplishes this feat of copying this end, even though it doesn't have the 3' hydroxyl group. And then I'll explain why some cells we want to die, and other cells we want to make sure we fix this end. But first, let's talk about how we fix this end chromosome problem. So I'm going to erase this here. So to describe how we're going to fix this end problem, let me first begin by drawing the chromosome. And I'm going to focus on this end here, the part with the overhang. Here we have our 3' here, and this is 5'. This, as we said before, is called the telomere, and it's going to have a repeating sequence of G, 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 so 4 G's, and then 2 T's. And then there's going to be 4 G's, 2 T's, 4 G's, 2 T's as you extend down the line. And the goal is to be able to extend this and copy it out. So to be able to insert an A, C, 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 C here. This is accomplished by a clever enzyme called telomerase. And what telomerase does is it will, as you'll see here, it will artificially extend this sequence here. So I'm going to redraw this here, and I intentionally gave myself a little bit more room here to work with. So telomerase will come in here, and it's a protein. In fact, why don't I write that here real quick? It's an enzyme that contains both protein plus RNA. And let me redraw our nucleotides here. G, 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 T, T. This enzyme, this telomerase enzyme, will bind this region here. That's the protein part of it. Within this protein now is RNA. And it has a very specific sequence, this RNA. As you might have guessed, A, C, 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 C. And then it extends it even further out. In fact, I'm going to erase this side here a little bit, and it'll go A, then C, 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 C. And let's write the whole protein encompassing it here. Now, by adding these extra bases to the top here, we've created a template that we can now take DNA polymerase and add to this. And so now we'll put new DNA here, T, T, G, 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 G. The same sequence that's here. So it's just making the telomere, the end of the chromosome, longer. Now I can't, because of the space here, I can't keep doing this. But what happens is this enzyme will now release itself, And so I can show you that. It's going to, this green enzyme, the telomerase here, is going to fall off. So let's go ahead and erase it. And as it falls off, it, of course, will take the RNA with it. And then what it'll do, and I'm not going to show this, you just need to know it, is it will reattach to this telomere it just added. And again, extend it by 6 more bases. And it will keep doing that until it has a very long extension here. So with this very long tail here, you might think, well, how does that fix the problem? Because we still have this overhang here, and it's still going to want to be sticky, and it's still going to want to loop up on itself or maybe attach to another chromosome or begin to degrade here. So what happens now with this really long strand here, I only had it extend 1 cycle, but remember, it's going to extend many, many times here. So you might be asking yourself, once we've added this tail here, this overhang, and remember, it's much larger than what I'm showing here, because the telomerase keeps binding and adding it much further downstream here, well off this whiteboard. So we have this very long tail. And so the question is, how does that protect the ends? Because still we have this overhang. And this overhang will be just as sticky as this original one. It will degrade and be unstable and bind to other ends of other chromosomes. So it's still a problem. But what we've created, or rather what telomerase has created, is this very long template. And with this very long template, our friend from the past here, RNA primase, adds an RNA primer. And remember, we're growing in the 5' to 3' direction. And so what it has done is artificially added this 3' hydroxyl group. And that is all DNA polymerase needs. Once DNA polymerase has this new primer with this new hydroxyl group added, it can begin to synthesize the DNA covering this original gap. Now, you might ask, isn't this primer going to be removed, and aren't we now going to still have this overhang problem? You're right, it does. So the primer is removed. However, it doesn't create as much of a problem now, because what we've done is have what's being removed here, because of it, the end being unstable, is the part that we just added. So you're really not shortening the chromosome, you're only shortening it by the amount you added. So this is the way that telomerase binds to the telomeres to prevent the chromosomes from shortening. Because remember, if it didn't do this, the chromosome would degrade this overhang, and each round of replication it would get shorter and shorter and shorter. This prevents that from happening. And it prevents the cell from dying. I think this is a very complicated process. So make sure you go through this again, read your textbook, and draw this over and over again. But now I want to come back to a final question here, and that is, do we always want to prevent the cell from dying? And I'm not going to answer this question right now. We might talk about this in class. We will talk about this in class. But I want to write down a couple of things here. Telomerase, remember that's the enzyme that binds to the ends, and it prevents cell death. So when there's no telomerase in a cell, the cells at some point, because as that overhang gets shorter and shorter and shorter, that will signal the cell to die. So with telomerase not being present, we will at some point cause something known as programmed cell death. Or you might have heard it before as apoptosis. So the question I want you to think about is, which cells in our body, let's think about humans, but any multicellular organism would work here, but which cells in our body do we want to be essentially, let's just say, immortal? Or rather, we don't want them to die prematurely. And you think of a disease linked to having too much telomerase present. Likewise, on the bottom here, when no telomerase is present, we do get programmed cell death at some point. Are there any cells in our body that we want to be mortal? Meaning we want them to die at some point. Can you think of any disease that might be linked to having no telomerase in cells that you might want to have telomerase present? We'll talk more about this tomorrow. These two questions that I just posed to you right now will not be on the quiz, so don't worry about like struggling trying to find an answer to them right now. But they will be important as we begin to discuss our activity tomorrow in class, and you will need to know for the exam as well. Okay, I'm going to go ahead and erase this and close with a quick summary. From this podcast, you should have learned some of the similarities between bacterial and eukaryotic, abbreviated here, replication, also abbreviated. You should have also learned about the differences between bacterial and eukaryotic DNA replication. Some of these differences included the number of origins of replication, in that eukaryotic cells have more origins of replication than bacteria, and the problems associated with that. Remember, we talked about the pre-replicating complex and how that signals replication to begin, and then we talked about that protein called gyminin and how that protein prevented multiple rounds of replication from the same origin of replication. Another difference was that there are more DNA polymerases in eukaryotic cells. And finally, we ended with talking about the chromosome ends, since they are linear chromosomes, and how we had to think about the telomeres and the enzyme telomerase. And I closed posing the questions of which kinds of cells do we want to make sure telomerase is always expressed in, and what kind of cells do we want to make sure it's not expressed in, which we'll talk about in class tomorrow. That ends this podcast. If you have any questions at all, please make sure you contact me, and I'll answer any questions you might have. I'll see you in class. Bye.